This micro lecture is on cell structure and function.
First, what is a cell? A cell is the smallest structural and functional unit of an organism. When we talk about a cell, we have to talk about Robert Hooke. He's the first person to have created and used a microscope. In doing so, he observed cork, and looking at the cork, he noticed that there were compartments within it, which he called cells. When we talk about cells, we have to talk about cell theory. There are three parts to cell theory. The first says that all organisms are comprised of at least one cell. All cells come from pre-existing cells, and the cell is the basic structural and functional unit of life. So what features do all cells have? They all have a plasma membrane. They all have cytoplasm. They all have genetic material, and they all have ribosomes. So what does a plasma membrane do? It regulates which molecules are allowed to enter and exit the cell. How does it do it? It is a selectively permeable membrane, and it's based on the electrical charge, the polarity, and to an extent the size of the molecule. Uncharged and very small lipid-soluble molecules can pass through the membrane more easily than larger polar molecules that carry a charge. A plasma membrane is made up of a phospholipid bilayer that forms a fluid mosaic. There are a number of anchored or free-floating membrane proteins contained within that fluid mosaic, and they can move around within it if they're free-floating. Different types of membrane proteins. We have transport, receptor, and recognition, and plasma membranes contain thousands of these. Transport proteins move molecules across the membrane. Receptor proteins deliver chemical messages and trigger a reaction inside the cell, and recognition proteins serve to identify cells. So how do molecules move in and out of the cell? Well, there's passive transport and there's active transport. Passive transport requires no energy. Active transport does. Gradients are one of the most important biological phenomena. If allowed, molecules will move down a concentration gradient until equilibrium is reached, i.e. diffusion. We're talking about things like solutions, obviously. A solution is a liquid mixture in which the solute is dissolved and uniformly distributed. The solute is the dissolved substance, and the solvent is what we're dissolving it into. So when we talk about diffusion, we're talking about things moving from high to low concentration. The stronger the concentration gradient, the faster the rate of diffusion, and diffusion will continue until the gradient no longer exists. You can think about dropping some food coloring into water and watching it spread out. Osmosis adds a selectively permeable membrane and is the diffusion of water across that selectively permeable membrane. When we talk about solutions, we have to talk about tonicity. In a hypertonic solution, the solution has a higher concentration of the solute than the cell does. And in this case, water would move out of the cell. You can see down in the graphic below, this is a blood cell. And assuming that the solution outside the cell is a higher concentration than the solution within, water moves out of it and you get crenation, which is the cell starts turning all spiky. A hypotonic solution has a lower concentration outside than inside. So in this case, outside the cell is a lower concentration of solute than is within the cell. In this case, as you can see down here on the lower right, the water would move into the cell, causing the cells to swell and eventually burst. What we want in our blood is an isotonic solution, where both the concentrations inside and outside the cell are equal, and the blood can look like its usual donut shape and do what it's supposed to. We also have to talk about passive transport in a little bit more detail because there's several different types. There is simple diffusion. There is facilitated diffusion, where channel proteins or carrier proteins assist, and there is osmosis. These require no energy, but they're types of transport. We also have endocytosis. where the cell membrane engulfs something from the outside and brings it to the inside. This is through forming a vesicle. There are three kinds. You have pinocytosis, which is cellular drinking, phagocytosis, which is cellular eating, and receptor-mediated endocytosis, which only takes in certain things that are identified by protein receptors. So having talked about that, then we have to talk about a little bit of the actual structure and function within cells. Common cell characteristics, they all have molecular components: proteins, amino acids, lipids, carbohydrates, sugars, nucleotides, RNA, DNA. They all have structural components, such as plasma membrane, cytoplasm, and ribosomes. And they all have some form of metabolism, which is getting energy and nutrients from the environment to build and repair cellular parts. So why are cells so small? They are small to minimize the ratio of surface area to volume. This allows for nutrients to come in and waste to move out of the cell more quickly. Diffusion is a slow process. If a cell had 4 inches of cytoplasm, it would take 200 hours for oxygen to diffuse across, which really wouldn't be good for us. So talking about different types of cells, you have prokaryotic and eukaryotic. Prokaryotic cells have a lot of the same things that a eukaryotic cell does, minus the membrane-bound organelles and the nucleus. They have a plasma membrane, they have cytoplasm, they have DNA, but instead of a nucleus, they have a nucleoid region, and they do have ribosomes, but they lack mitochondria or Golgi bodies or endoplasmic reticulum. Prokaryotic cells are a much simpler structure. They generally lack membrane-bound organelles. They may have some organelle-like structures. They have a singular circular strand of DNA that's coiled and attached to the plasma membrane and located in the nucleoid region. Some of them have a cell wall, and they divide by binary fission. Eukaryotes are primarily distinguished by the presence of nucleus and other membrane-bound organelles. So that leads us to the question of what is an organelle? An organelle is a specialized structure within a cell. The membrane that bounds them allows those organelles to confine enzymes, reactants, and proteins away from the rest of the cell. We talk about eukaryotes, we have to talk about the two different types. We have plants and we have animals. So how are they different? Well, they're different in the shape. Plants have a cell wall, while animal cells do not. They just have a cell membrane, meaning that plants are more rectangular and rigid in their cells versus round, widely varying animal cells. Their internal arrangement is a little bit different. We have a few different organelles. We don't have chloroplasts. Animal cells don't have chloroplasts, whereas plant cells do. Plant cells have an offset nucleus that is not directly in the center of the cell, while animal cells have their nucleus centralized. And of course, metabolism, autotroph versus heterotroph. An autotroph can make their own energy. Plants do that using chloroplasts to generate energy from the sun. Heterotrophs have to take in their energy from other sources, so we have to consume other things. So talking about specific organelles and their functions, this is just looking at the difference between plant and animal cells. You can see the central vacuole in the plant cell, which animal cells don't exactly have. You can also see the cell wall and the chloroplasts within it. And then on the animal side, you can see that we don't have the cell wall, and while we have the rest of it, we also don't have chloroplasts. So the nucleus is the brain of the cell. It contains DNA. DNA is chromatin, which is condensed into chromosomes. That's what the nucleotides make up the chromatin. Chromatin then condenses into chromosomes, and that contains all of our blueprints for everything that our cells do and everything that they are. Then you have the nuclear envelope, which is 2 membranes that holds that DNA into a centralized location. And you have the nucleolus, which synthesizes ribosomes. Now, ribosomes parts are made in the nucleolus. They're assembled in the cytoplasm, and they function as the workbench of the cell by doing protein synthesis. So those are what synthesizes proteins for us. We also have the endoplasmic reticulum, The endoplasmic reticulum comes in two types. Rough endoplasmic reticulum has embedded ribosomes and synthesizes proteins, and smooth endoplasmic reticulum, which synthesizes lipids and other molecules. Both generate vesicles that transport products. The Golgi apparatus acts as the transportation hub, where vesicles are received, repackaged, and sometimes modified before being sent to their final destination. The Golgi apparatus makes glycoproteins, which are proteins with sugars attached. We often use those as recognition proteins on cell membranes. We also have something we call lysosomes. Lysosomes are specialized vesicles created in the Golgi apparatus that contain digestive enzymes. They act as recycling centers for the cell. And when we talk about energy production and consumption, only plants, autotrophs, can produce their own energy. They do that with photosynthesis, which is literally synthesis from light. Photosynthesis is complementary to respiration. And that's done in the mitochondria. Mitochondria breaks down sugars into energy. And like chloroplasts, they're a membrane-rich structure. We'll talk more about those as we go further into talking about photosynthesis and respiration. Now, here's something interesting about chloroplasts and mitochondria. They both possess their own DNA. They may replicate independently of the cell, in conjunction with the cell, or depending on metabolic needs. And they are thought to have originated as bacteria that were engulfed by eukaryotic cells, making them endosymbionts. Last thing we have to talk about is the cytoskeleton. The cytoskeleton is a network of protein fibers attached to the organelles. They control and help regulate the shape of the cell, the movement of the cell, the movement of the organelles within the cell, and cell division. Without them, everything would just be completely free-floating and would change depending on motion or orientation of the body. And that's not really good for us, so the cytoskeleton helps keep that from happening. That's your biology bite for the week, talking about the high points of what you should be getting out of the lecture. I hope it was helpful, and I will see y'all next week.